Dear learners, in this video we are going to study unit 6 reproduction. Chapter 1 is sexual reproduction in flowering plants. Chapter 2 is human reproduction. Chapter 3 is reproductive health. So these three chapters are we are going to cover in this unit. In this video we will cover this first chapter. Biology, in essence, is the story of life on Earth. While individual organisms die without fail, species continue to live through millions of years unless threatened by natural or anthropogenic extinction. Reproduction becomes a vital process without which a species cannot survive for a long. Each individual leaves its progeny by asexual or sexual means. Sexual mode of reproduction enables creation of new variants so that survival advantage is enhanced. This unit explains the details of reproductive processes in following plants and flowering plants and human as easy to relate representative examples. A related perspective on human reproductive health and how reproductive ill health can be avoided is also presented to complete our understanding of biology of reproduction. Coming to the next piece, the in which we have given about Panchanan Maheshwari. He was born on 1904 November and died on 1966. So here study about it. Born in November 1904 in Jaipur, Rajasthan, Panchanan Maheshwari rose to become one of the most distinguished botanists not only of India but of the entire world. He moved to Allahabad for higher education, where he obtained his DSc during his college days. He was inspired by Dr. W. Dudgeon, an American missionary teacher, to develop interest in botany and especially morphology. His teacher once expressed that if his student progress ahead of him, it will give him a great satisfaction. These words encouraged Panchanan to inquire what he could do for his teacher in return. He worked on embryological aspects and popularized use of embryological characters in taxonomy. He established the Department of Botany, University of Delhi, as an important center of research in embryology and tissue culture. He also emphasized the need for initiation of work and artificial culture of immature embryos. Sorry. These days, tissue culture has become a landmark in science. His work on test tube fertilization and intra-ovarian pollination won worldwide acclaim. He was honored with Fellowship of Royal Society of London (FRS), Indian National Science Academy, and several other institutions of excellence. He encouraged general education and made a significant contribution to school education by his leadership in bringing out the very first textbook of biology for higher secondary schools, published by NCERT in 1964. Coming to the chapter 1, that is sexual reproduction in flowering plants. Here we study, are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. At a look at diversity of structures of the inflorescences, flowers and floral plants shows an amazing range of adaptations to ensure formation of the end of products of sexual reproduction. The fruits and seeds in this chapter, let us understand the morphology, structure, and the processes of sexual reproduction in flowering plants. Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. So this is a question. Next is Find out the names of five more flowers that are used in social and cultural celebrations in your family. Have you heard of floriculture? What does it refer to? So answer this question. Biologists, flower are morphological and embryological marvels and of sexual sites of sexual reproduction. In early classes, you will you have to read the various parts of a flower. Figure 1.1 will help you recall the parts of a typical flower. Can you name two parts in a flower in which the two most important units of sexual reproduction develop? Pre-fertilization structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated, which led to the differentiation and further development of floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and gynecium differentiate and develop. You would recollect that the androecium consists of whorl of stamens representing the male reproductive organ and the gynecium represent the female reproductive organ. Stamen, microsporangium, and pollen grain. Figure 1.2a shows the two parts of typical stamen, the long and slender stalk called filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or petal of the flower. The number of length of stamens are variable in flowers of different species. If you were to collect a stamen each from 10 flowers and arrange them on a slide, you will be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having toothpica. It means they are diathesious. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various type of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct and transverse section of the anther. The anther is four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobes. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangium in a transverse section of typical microsporangium appears near circular in root outline. It is generally surrounded by four wall layers: the epidermis, endothecia, middle layers, and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing uh, pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. Microsporogenesis: as the anther develops, the cells of sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or a microspore mother cell. The process of formation of microspores from a pollen mother cell (PMC) through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anther mature and dehydrate, the microspores dissociate form to each other and develop into pollen grains. Figure 1.3b. Inside each microsporangium, several thousand of microscope or pollen grains are, grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grains are represent the male gametophytes. If you touch the open anther of hibiscus or any other flower, you will find deposition of yellowish powdery pollen grains on your fingers. Sprinkle Sprinkle these grains on a drop of water taken in a glass slide and observe under microscope. You will really be amazed at the variety of architecture, size, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has prominent two-layered wall. The hard outer layer is called exine and is made up of the sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent apparatus called germ pores. When sporopollenin is absent, pollen grains are well preserved as fossil because of presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of pollen grain is Pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and nucleus. In over 60% of angiosperms, pollen grains are shed at this two-celled stage. In the remaining species, the generative cell divides mitotically to give rise the two male pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass cat that came to India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablet as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen's consumption has been claimed to increase the performance of athletes and race horses. When one they are shed, pollen grains have to land on stigma before they lose viability of they if they have to bring about fertilization. How long do you think the pollen grain retain viability? The period for which pollen grain remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, Leguminosae and Solanaceae, they maintain viability for months. You may have heard of storing semen or sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen. Such stored pollen grain can be used as pollen banks, similar to seed banks in crop breeding programs. The pistil, megasporangium, and embryo sac. The gynoecium represent the female reproductive part of the flower. The gynoecium may consist of a single pistil monocarpellary or may have more than one pistil multicarpellary. carpellary. When there are more than one, the pistils may be fused together, syn- uh, syncarpus, or maybe free apocarpus. Each pistil has three parts: the stigma, style, and the ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in class 11th. Arising from the placenta are megasporangia, commonly called ovules. The number of ovules in the ovary may be one, wheat, paddy, mango, to many papaya, watermelon, orchids. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of a ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between the ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments in our encircle the nucellus except at the tip where a small opening called micropyle is organized. Opposite the micropylar and is calza representing the basal part of the ovule. Enclosed within the integuments has a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials located in the nucellus. Is the embryo sac or female gametophyte? An ovule generally has single embryo sac formed from a megaspore. Megasporangenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of MMC undergoing meiosis? Meiosis results in the production of four megaspores, female gametophyte. In a majority of flowering plants, one of the megaspores is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. What will the ploidy of the cells of the nucellus, MMC, the functional megaspore and female gametophyte? Let us study about the formation of embryo sac in detail. The nucleus of the functional megaspore divides mitotically to form two nuclei, which move to opposite poles, forming two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of four nucleate and later eight nucleate stages of the embryo sac. It is of the interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. After eight nuclear stages, cell walls are laid down, leading to the organization of typical female gametophyte or embryo sac. Observe the, observe the distribution of cells inside the embryo sac. Six of the eight nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the last central cell. There is a characteristic distribution of cells within the embryo sac. Three cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have a special cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into synergid. The three cells are that the chelazal and and are called antipodals. The large central cell, as mentioned earlier, has two polar nuclei. Thus, typical angiosperm embryo sac at maturity through 8 nucleate is 7 celled. Pollination. In the preceding sections, you have learned that the male and female gametes in the flowering plants are produced in the pollen grain and embryo sac respectively. As both type of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains said from the anther to the stigma of a pistil termed pollination. For all flowering plants have involved an amazing array of adaptation to achieve pollination. They make use of external agent to achieve pollination. Can you relate the possible external agents? Kinds of pollination. Depending on the source of pollination, pollination can be divided into three types: autogamy, geitonogamy, xenogamy, and then agents of pollination. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from anther to stigma of the same flower. In a normal flower which opens and exposes the anthers and the stigma, complete autogamy is rather rare. Autogamy in such flower requires synochrony in pollen release and stigma respectively, receptivity. And also the anthers and the stigma should lie close to each other so that the self pollination can occur. Some plants such as viola, common pansy, oxalis, and Camelina produce two types of flowers: Casmogamous flowers, which are similar to flowers of other species with exposed anther and stigma, and Clistogamous flower, which do not open at all. In such flower, the anther and stigma lie close to each other. When anther dehisce in the flower buds, pollen grain come in contact with the stigma to affect pollination. Then, thus, Clistogamous flowers are invariably autogamous, as there is no chance of cross pollen landing on the stigma. Cleistogamous flowers produce azure seed set even in the absence of pollinators. Do you think that Kleistogamy is advantageous or disadvantageous to the plant? Why? Getinogamy. Transfer of pollen grains from the anther to the stigma of another flower is the same plant. Although geitonogamy functionally cross pollination involving a pollinating agent genetically, it is similar to autogamy since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grains from anther to stigma of a different plant. This is only type of pollination during pollination, which during pollination brings genetically different type of pollen grain to the stigma. Agents of pollination. Plant use two abiotic wind and water and one biotic animal agent to achieve pollination. Majority of plant use biotic agent from pollination. Only a small proportion plant use abiotic agents. Pollen grain coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grain, the flower produces enormous amount of pollen when compared to number of ovules. When compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollinations. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transported in wind current. They often possess well-exposed stamens so that the pollen are easily dispersed into wind currents, and large often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A friendly example is the corn cob. The ears you see are nothing but the stigma and the style, which have wave in wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plant and then limited to about 30 genera, mostly monocotyledon. As again this you would recall that water is a regular mode of transport for male gamete among the lower part plants such as algae, bryophyte, teratophyte. It's believed particularly for some bryophytes and teratophyte that their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some example of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water and Several marine sea grasses, such as Jostera, not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insect or wind, as in most of the land plant. In Vallisneria, the female flower reach the surface of water by the long stalk, and the male flower of or pollen grains are released onto the surface of water. They are carried passively by water currents. Some of them eventually reach the female flowers and the stigma. In other group, the water pollinated plants such as sea grasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them release stigma and achieve pollination. In most water pollinated species, pollen grains are protected from wetting by a वो mucilaginous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? Majority of flowering plants use a range of animals as pollinating agents: bees, butterflies, flies, beetles, wasp, ant, moth, bird, sunbird, and hummingbird. And bats are common pollinating agents. Among the animals, insect, particularly bees, are the dominant biotic pollinating agent. Even larger numbers such as some primates, lemurs, arboreal, tree-dwelling rodents, or even reptile, gecko, lizard, and garden lizard have also been reported as pollinators in some species. Often, flowers of animal pollinated plants are specifically adapted for a particular species of animal. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flowers by color or by fragments. The flowers pollinated by flies and beetles secrete full order to attract these animals. To sustain animal visit, the flower have to provide a reward to the animal. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitor comes in contact with the anthers and the stigma. The body of animal gets a coating of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body come in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe places to lay eggs. An example is that of tallest flower of Amorphophallus. The flower itself is about 6 feet in height. A similar relationship exists between a species of moth and plant yucca, where both species, moth and plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larva of moth come out of eggs, and the seed start developing. Why don't you observe some flowers of following plant or any other available to you: cucumber, mango, people, coriander, papaya? Coriander, papaya, onion, lobia, tobacco, rose, lemon, eucalyptus, and banana. Try to find out which animals visit them, and whether they could be pollinators. You will have to patiently observe the flower over a few days, and at different times of the day. You could also try to see whether there is any correlation in the characteristic of a flower to the animal that visits it. Carefully observe it. Any of the visitors came in contact with the anther and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or nectar without bringing about pollination. Such flower visits are referred to as pollen or nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your efforts. Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is incompatibility. This is genetic mechanism and prevents self-pollen from the same flower to or other flower of the same plant from fertilization by the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent the self-pollination is the production of unisexual flowers. In both male and female flowers are present on the same plant such as castor and maize, monoecious. It prevents autogamy but not gitonogamy. In several species such as papaya, male and female flowers are present on different plant. That is, each plant is either male or female. This condition prevents both autogamy and gitonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as the stigma, often pollen of the wrong type either from either species or from the same plant. If it is self incompatible, also land on the stigma. The pistil has the ability to recognize the pollen whether it is of the right type or of the wrong type. If it is right type, the pistil accept the pollen, promotes post pollination events that leads to fertilization. If the pollen is of the wrong type, the pistil reject the pollen by preventing pollen germination on the stigma or by pollen or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of the continuous dialogue between pollen grains and pistil. That this dialogue is mediated by chemical component of the pollen interacting with those of pistil. It is only in the recent year that botanists have been able to identify some of the pollen and pistil components and in the interaction leading to the recognition or followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pore. The content of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style, which is the ovary. You would recall that in some plants, pollen grains are set at two celled condition, a vegetative cell and generative cell. In such plant, the generative cell divides and forms two male gamete during the growth of pollen tube in the stigma. In plants which set pollen in three celled condition, pollen tubes carry the two male gamete from the beginning. Pollen tube, after reaching the ovary, enter the ovule through micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropyle part of the synergids guide the entry of pollen tube. All these events, from pollen deposition on the stigma until pollen tube enter the ovule, are together referred to as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion followed by the promotion or inhibition of the pollen. The knowledge gained in this area is would help the plant breeder in manipulating pollen pistil interaction in even in incompatible pollination to get desired hybrids. You can easily study pollen germination by testing some pollen from flowers such as pea, chickpea, Crotalaria balsam and vinca on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of pollen grains. A breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approaches to crop improvement program. In such a crossing experiment, it is important to make sure that only desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted plant. This is achieved by emasculation and bagging techniques.
If the female parent bear bisexual flowers, removal of anther from the flower bud before the anther dehisces using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of black flower contains receptivity, mature pollen grain collected from the anther of the male parent are dusted on the stigma and the flowers are rebagged and thus fruits allowed to develop. In the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Double fertilization. After entering one of the synergids, the pollen tube releases two male gametes into cytoplasm of the synergids. One of the male gametes move toward the egg cell and fuses with its nucleus, thus completing the syngamy. This results in formation of a diploid cell zygote. The other male gamete move toward the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus (PEN). As this involves a fusion of three haploid nuclei, it is termed as triple fuse. Since two type of fusion, syngamy and triple fusion, take place in the embryo sac, the phenomena is termed as double fertilization. An event unique to flowering plant, the central cell after triple fusion becomes primary endosperm cell (PEC) and develops into the endosperm, while zygote develops into an embryo. Post fertilization. These are the diagrams for showing fertilized embryo sac, showing zygote and primary endosperm nucleus. These are the stages in the embryo development in a dicot. So these are shown in reduced diagram as compared to A. Post fertilization structures and events. Following double fertilization events of endosperm and embryo development, maturation of ovules into seed and ovary into fruit are collectively termed post fertilization events. Endosperm development precedes embryo development, while the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cell of this tissue are filled with reserved food material and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with, the nothing but free nuclear endosperm made up of thousands of nuclei. And the surrounding white kernel is the cellular endosperm. Endosperm may be either of completely consumed by developing embryo, for example, pea, groundnut, beans before seed maturation, or it may persist in the mature seed castor and coconut and be used up during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruit of coconut and look for the endosperm in each case. Find out whether endosperm is persistent in cereals, wheat, rice and maize. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though, though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure 1.13 depicts the stages of embryogeny in a dicotyledonous embryo. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped, and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. And two cotyledons.
The portion of embryonal axis above the level of cotyledons is the epicotyl, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyl, that terminates as its lower end in the radicle or root tip. The root tip is covered with a root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side, lateral of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment to the of scutellum is the epicotyl. Epicotyl has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe the various parts of the embryo and seed. Coming to the next heading, that is seed. An angiosperm, in angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of a seed coat or coats, cotyledon or cotyledons, and embryo axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to the storage of food reserves. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. Example, pea, groundnut, albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. Examples are wheat, maize, barley, castor. Occasionally, in some seeds, such as black pepper and wheat, remnants of nucellus are also persistent. This residual persistent nucellus is the perisperm. Integuments of the ovules harden as tough protective seed coats. The micropyle remains as a small pore in the seed pod. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry. 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. As ovules mature into seeds, the ovary develops into a fruit. It means the transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. The fruits may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard, etc. Many fruits have evolved mechanisms for dispersal of seeds. Recall classification of fruits and their dispersal mechanisms that you have studied in an earlier classes. Is there any relationship between number of ovules in an ovary and the number of seeds present in the fruit? In the most plants, by time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to the fruit formation. Such fruits are called false fruits. Most fruits, however, develop only from ovary and are called tofu. Although in most of the species, fruits are the results of fertilization, there are few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpy can be included through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats and help the species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard sheath coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combinations, leading to variations. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds is crucial for the storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seeds or in the presence of seeds which germinate and straight away soon after formation and cannot be stored? How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, seeds lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seed. The oldest is that of Lupin. Lupinus arcticus excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2000 years old viable series of date palm, Phoenix dactylifera, discovered during the archaeological excavation at King Herod's place near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following question: how many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one of the such category, and each fruit contains thousands of tiny seeds. Similar is the case of in fruits of some parasitic species such as Orobanche and Striga. Have you seen a tiny seed of ficus? How large is the tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? Apomixis and polyembryony. Although seeds in general are the products of the fertilization, a few flowering plants and such as some species of asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? This apomixis is a form of asexual reproduction that mimics a sexual reproduction. There are several ways of development of apomictic seeds. In some species, diploid axil is formed without reduction division and develops into the embryo without fertilization. More often, as in citrus, many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such a species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Hybrid varieties of several of the food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly, and hence the cost of hybrid seeds become too expensive for the farmers. If these hybrids are made into apomicts, there is no segregation of characters in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in the hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. Coming to the summary, coming to the summary, flowers are the seat of asexual reproduction in angiosperms. In the flower, androecium consists of stamens, represent the male reproductive organs, and gynoecium, consisting of pistils, represents the female reproductive organs. A typical anther is bilobed, diathesis, diathesis, and tetrasporangiate. Pollen grains develop inside the microsporangia, four wall layers, the epidermis, endothecium, middle layers, and the tapetum surround the microsporangium. Cells of sporogenous tissue lying in the center of the microsporangium undergo meiosis to form tetrads of microspores. Individuals microspores mature into pollen grains. Pollen grains represent the male gametophytic generation. The pollen grains have two layered wall, the outer exine and the inner intine. The exine is made up of sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and an generative cell, or three cells, a vegetative cell and two male gametes. At the time of shedding, the pistil has three parts: the stigma, style, and the ovary. Ovules are present in the ovary. Ovules have a stalk called punicle, protective integuments, and an opening called micropyle. The central tissue is the nucellus in which the archisporium differentiates. A cell of the archaeosporium, the megaspore mother cell, divides meiotically, and one of the megaspores forms the embryo sac, the female gametophyte. The mature embryo sac is seven celled and eight nucleate. At the micropylar end is the egg apparatus consisting of two synergids and an egg cell. At the chelazal and are three antipodals. At the center is a large central cell with two polar nuclei. Pollination is the mechanism to transfer pollen grains from the anther to stigma. Pollinating agents are either abiotic, wind and water, or biotic animals. Pollen pistil interaction involves an event of landing of pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen emission when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma and resulting pollen tube growth through style enter the ovules and finally discharges into male and male gametes in one of the synergids. Angiosperms exhibit double fertilization because two fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus. In the primary endosperm cell, zygote develops into the embryo, and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo, globular, and heart-shaped stages before maturation. Mature dicotyledonous donous embryo has two cotyledons and an embryonal axis with epicotyl and hypocotyl. Embryos of monocotyledons have a single cotyledon after fertilization. Ovary develops into fruits and ovules develop into seeds. A phenomena called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomix has several advantages in horticulture and agriculture. Some angiosperm produce more than one embryo in their seed. This phenomenon is called polyembryony. So the chapter is finished. Thanks for